And I'm going to redraw this again. So can I erase this too? Okay. All right, so we talked about the blood supply into the kidney. And to go back to, this diagram, we have this intimate relationship between the glomerulus and epithelial cells forming the capsule and our kidney tubule. That functional unit that does the filtration and modifies the content of the filtrate is called a nephron. Okay? Then when the nephron has done its function, that filtrate drains into a structure known as a collecting duct. And that is where we modify the amount of water that's in the filtrate. And then the collecting duct actually empties into our calyx. Okay? So let's redraw this and add in our tubule. So again, this is the cortex. This is the medulla. Okay. We would have here an interlobar artery. This would be the arcuate artery. And the interlobular artery. So as blood comes in, it comes up the interlobar artery, goes through the arcuate artery, comes up the interlobular artery, and branching off of the interlobular artery, we have the afferent arterioles. Then the afferent arteriole loses its smooth muscle layer, and we have the glomerulus. Which, if you want to, is also known as glomerular capillaries. So the glomerulus is just this ball or bud of glomerular capillaries. Then the capillaries come back together to form a single vessel again. Known as the efferent arteriole. A bit unusual to have an arteriole and a capillary bed and then another arterial. Here, the efferent arterial is going to then lose its smooth muscle again to form peritubular capillaries. Okay. So on our diagram, here is the afferent arterial, here is the glomerulus, here is the efferent arterial, here is our peritubular capillaries. Let me add an additional term. The glomerulus is capillaries, right? I just said glomerular capillaries. Cap, bed, 1. Sound familiar? Peritubular capillaries, cap, bed, 2. What does that remind you of? Hypothalamus, hypophyseal, portal system. And that system, in the hypothalamus and pituitary gland, we had arteries coming into the hypothalamus, forming cap bed 1, veins going from the hypothalamus down the pituitary stalk to the anterior pituitary, forming cap bed 2, and then veins leaving cap bed 2 and carrying the hormones away. What was the overall purpose for having 2 beds of capillaries? What happened in cap bed 1? 2 things. Hormone levels, such as estrogen or progesterone or thyroid hormone, left the capillaries and went into the hypothalamus where receptors could see what level those hormones were at. What did the hypothalamus add to the blood in cap bed 1? To be sent to cap bed 2. Releasing factors, right? So, or inhibitory factors if we're talking about dopamine and prolactin, but typically thyroid stimulating hormone, corticotropic stimulating hormone. Those went to the second capillary bed. Because there was a second capillary bed, we avoided having to go all the way to the heart and back again. And in the second capillary bed, those releasing factors entered into the anterior pituitary and secreted hormones were picked up. All right? We call that a venous portal system. And we will see that one more time when we get to the digestive tract, where a broken down, enzymatically broken down amino acids and glucose will be picked up by the capillary beds in the small intestine, transported to the liver, and leave the capillary beds in the liver to be utilized by the liver cells in synthesis or storage of starch. Now, this is similar. We have 2 capillary beds, but what's the difference? In the pituitary, in the digestive system, we have vein, artery, vein, capillary, vein, capillary. Here we have artery, arterial, capillary, arterial, capillary, vein. So this is known as an arterial portal system. What advantage or function does it have? Why would we, obviously we're going to take out particles in the first capillary bed. In the second capillary bed, the peritubular capillaries, we're going to reabsorb those particles that the body wants to keep. All the glucose, all the amino acids, 80% of the sodium ions. We're going to reabsorb back into the body. So why not just do veins? What is the unique function of arterials? Exactly right, blood pressure. Blood pressure is important in filtration. So, specifically right here, in the first capillary bed, by having an arterial on either side, we can control the filtration pressure within that first capillary bed. All right, we'll talk about this further on Tuesday. When the blood pressure is too high, and we're in danger of damaging that filtration membrane, this first arteriole will constrict, and the second arteriole will dilate, and that will lower the filtration pressure. If blood pressure is too low, the reverse happens. The efferent arteriole will constrict, the afferent arteriole will dilate, and pressure across the filtration bed will increase. That's why we have arterioles on either side of that first capillary bed. Okay? The second capillary bed, yes, is functional and reabsorption of that material. But the reason we have two sets of arterioles is for that regulation. I had to smile a few years ago. I was redoing some of the questions for the take-home on the urinary, and I was looking some stuff up on the internet, and I came across, I don't know, it was Yahoo Answer or Answer.com or something, and there's an email from one of my students. Help! I have these questions on my exam, and there was a list of about 15 questions from one of their take-home exams, and one of them was, identify the type of portal system in the kidney. Venous portal system, arterial portal system. And the first answer that came back was, there is no portal system in the kidney. I was like, uh-oh. And the student wrote back, well, thanks for your help, but I already had to turn my test in. And, well, it's a good thing, because if you'd gone by the answers on the internet, you would have gotten it wrong, because there definitely is a portal system in the kidney. And it's a renal portal system rather than the venous portal system that we typically can think about. Okay?
You said venal?
Renal. The renal portal system.
So it's not arterial?
It's an arterial rather than venous. So we have arterioles on both sides of the first capillary bed rather than veins on both sides of the second capillary bed. Okay? Yes?
So renal, arterial, portal system?
Renal, arterial, yeah. So just real briefly, if you remember, in the hypothalamus, we had veins, cap bed 1. In the hypothalamus, veins again, the portal veins going down the pituitary stalk, cap bed 2 in the pituitary gland, and then pituitary veins carrying away the hormones back to the heart for distribution. Okay? So this is a venous portal system. And the same pattern we'll see again in the digestive system, where the veins of the small intestine going, oops, I'm sorry, did this wrong. Arteries. So arteries coming into the pituitary hypothalamus will form the capillaries, and then veins, and then capillaries, and then veins. And in the digestive system, arteries going to the small intestine will form capillaries where those lacteals were that we talked about. That will pick up the amino acids and the glucose. They will go from veins to the liver, and then into capillaries again of the liver, which we call sinusoids because they're really much larger in diameter, and then back into the veins, the hepatic veins, which leave the liver. So those two systems are very similar. In the kidneys, we have arteries. Obviously, we have arterioles here, but I'm going to write them in here. And then instead of, we have our capillaries. So that would be the glomerulus. Okay? And then instead of a vein, we would have the efferent arteriole. So here would be the afferent. Here would be the efferent. And then we have cap bed 2, which are the peritubular capillaries. And then we would go to veins. So here we have veins on either side of the second capillary bed. Here we have arterioles on either side of the first capillary bed. Okay. All right, so now let's look at the nephron. The nephron is defined as the functional unit of the kidney. So this is where we filter from the glomerular capillaries the contents that are going to be modified to form urine. Again, that process is what we're going to start talking about on Tuesday next week. So, this tubule is really, the nephron, is really a long tube. All right? Can I erase this portal system stuff? You guys, wait a second. Yeah, it's fine. Question?
I understand the sequence, but I seem to be missing the location where they actually.
All right, so first capillary, we have artery,
Yeah.
afferent arterial. First capillary bed is the glomerular capillaries, then efferent arterial. Second capillary bed are the peritubular capillaries. All of this here are the purple are the peritubular capillaries, and then that's going to get back into veins.
Oh, okay. I'm sorry. I just misunderstood the drawing. Then I wasn't seeing the connection
Yeah.
between the two sides. That's all.
Okay.
Yeah.
All right. So the nephron, I am going to draw in black. And essentially the nephron is the capsule. It's either called the glomerular capsule, or the older name is Bowman's capsule. And this is formed around the, I'll put it in a minute, I'll put it in this drawing over here. But this is where we separate by size small particles. This is where we actually create the filtrate. All right, so anything that's small enough to pass out of the blood, across the blood vessel, the capillary simple squamous epithelium, and across the simple squamous epithelium that forms our capsule is considered to be filtered. If it's too big, it doesn't cross. All right? But glucose, amino acids, hydrogen ions, sodium ions, certainly water molecules, are small enough to pass across this membrane. And on Tuesday, we'll talk about the actual structure, the cells that form this membrane. So it's similar to the sandwich that we described for the respiratory membrane, where we had two epithelia and a little bit of connective tissue in between. So with our glomerular filtration membrane, we have two epithelia, the epithelium of our capillary and the epithelium of our capsule. Okay? And then it goes into a tube, and again, this is just a schematic, which is the proximal meaning closest to the point of origin, which is the capsule, convoluted tubule. And I'll show the convolution over here. Right now we're just doing kind of an overall schematic, right? So we have a proximal convoluted tubule. This is where we start changing the content of the filtrate. As soon as it's in our capsule, it's now called filtrate. Not urine yet. We still have to modify it quite a bit. Okay? So in the proximal convoluted tubule, we will reabsorb material. So reabsorption occurs. Reabsorption means taking out of that filtrate. To illustrate, my kids are 20 and 15 now, so it's a little different from when they were 5 and 10. But they were 3 and 8 when we moved into the house we live in now, and all their bedrooms are down one wing of the house. The hallway that goes by their doors and in the bathroom. My husband calls it the Hall of Weasels. And they can have all sorts of stuff when they were little. Pencils, Legos, you know, bionic pieces, and they would just add to it. They would leave their backpacks outside their door, so they didn't have room in their bedrooms to take it into, and their shoes, and it's like the next day they would just, instead of picking something up and putting it away, they would just walk over it. Wouldn't even see that it was there, okay? So let's say that I get tired of this mess, and so I finally have spring break, or the first day of summer. Okay, make it the second day of summer. First day of summer, I just class. Second day of summer, I said, okay, I'm going to clean house. And so I get a big box, and I never put away a single Lego piece in my life. I threw it away, okay? Which apparently they're collector items now, so I don't know how much money I threw away, but I didn't buy Legos. My sister-in-law gave 2 big bags to us, which I've now given to her daughter and said, have fun. And let's say I just go through the hallway, the family room, and everything I found that fit into this big box, I picked up and I put in the box. iPods, Lego pieces, you know, whatever, toys, dolls, whatever, Bionicles, put them in there if they fit. Hence, filtration by size. Carried it out to the garage, and I'm going to throw it away, all right? After 2 or 3 days, the kids finally notice that something's missing. And it's like, Mom, I need whatever. I'm like, okay, if you want it, it's out there in the garage. So they go back out to the garage, and they start taking out of that box the stuff that they want to keep. Reabsorption, okay? And I continue to find stuff that they're not putting away, and I take it out to the garage, and I add it to the box. Kids, secretion, all right? So in the proximal convoluted tubule, this is where we take all the glucose back, all the amino acids. 80% of the sodium is returned back to the blood, the peritubular capillaries. That's its purpose, okay? Now, another segment, the secretion is usually hydrogen ions, okay? Or if you take antibiotics, the products of breaking down those drugs can be added back. There is a very thin segment of the nephron known as the nephron loop. And this is used to set up a salt gradient in the medulla, so that it's essentially the same saltiness as blood right up here, but as we move farther and farther closer to the papilla of the pyramid, it can actually get to as high as 1,400 milliosmoles, almost 5 times the number of particles in the blood. Okay, and we're going to talk about how this happens on Tuesday. So, suffice it to say, I'm just going to write osmotic gradient. Then, at the end of the nephron is another segment known as the distal convoluted tubule. And this is where we do reabsorption again, and secretion again, but regulated. This is where aldosterone works. So if we need to take more sodium out, aldosterone is going to work on the distal convoluted tubule. Okay? And that's the nephron. We haven't yet finished it because we haven't done the concentration, but that's essentially the function of the nephron. Okay? And now I'm going to take that nephron and put it in place with our blood supply. So on Tuesday, we're going to start with the capsule. I'm going to show you how the cells are layered and how they work together, and talk about how the glomerular filtration pressure is determined and regulated. We'll look at the cells of the convoluted tubule and the nephron loop. Okay? We'll save the distal convoluted tubule for when we talk about acid-base electrolyte regulation. That will be our content for lecture on Tuesday. So, now let's put this into place. So the capsule contains this tuft of capillaries. The inner layer is intimately associated with these blood vessels. In other words, if I were to take a ball of string, all kind of rounded around, and dip it into melted plastic and pull it back out, that's how that inner layer wraps around the capillaries to actually form the filtration membrane. Okay? So it's much more intimately than just pushing your fist into a balloon, because it completely surrounds each of the vessels. And then the outer layer comes like this. So this is our capsular space. This is the first place that the filtrate is going to enter. Again, I'm not going to go into too much detail because we'll talk about it on Tuesday.
And what are the black strings?
The black strings are actually the inner layer, the inner lining here. It's just more complicated than that because they're intimately wrapped around the blood vessels. Okay? So this would now be the beginning of our proximal convoluted tubule. So it's highly branched. And that just provides a greater amount of surface area for all of this to occur. Okay. So that proximal convoluted tubule is all of this. Now, if it's a nephron that's quite a ways from the arcuate artery, its nephron loop may not dip down into the medulla, and it's going to function just in that reabsorption secretion. If it's a nephron that's close to the arcuate artery, then its nephron loop is going to descend deep down into the medulla and come back up again. Okay, so this would be the nephron loop. And then it's going to come it's going to pass right between the afferent arteriole and the efferent arteriole. And that's very important because there are cells here that are sensitive to how many sodium ions are in this filtrate. And remember, the distal tubule is where we regulate ion concentration, so that's how the nephron is sensitive to how many sodium ions are present. Many of your diagrams, I'll get to you in just a second. Many of the diagrams, the artists were lazy. And so they will do this. Here's the proximal convoluted tubule. Here's the nephron loop. Here's the distal tubule. This is not anatomically correct. This part should be right back up here. Okay, but for whatever reason, clarification or make it easier to put space in there, they'll have it far away from there. It actually comes back right up through between the two. Okay? You got a question?
So the vasa recta and the nephron loop are two different things.
Yes, they're related functionally, but the vasa recta are blood vessels. The nephron loop is part of the nephron. Just really quickly, remember I said the nephron loop makes this concentration gradient? So I kind of have to talk about stuff I haven't explained yet, but I made strawberry shortcake for my family the first time Monday nights for this season, okay? Before I served it, about half an hour before I served it, after my husband cut up the strawberries, I added sugar. By the time I served it, I had strawberry juice, okay? Because that sugar automatically pulled water out of the strawberry cells. So, this gradient, mostly of sodium ions, but some other components as well, is a higher osmolality than the filtrate coming in. This is where ADH will work. If the body is low on water, you haven't drunk for a day, water, not alcohol, haven't drunk for a day, then ADH will cause water channels to be added to these cells. And as the filtrate passes, and it's this medulla that makes that, as the filtrate passes down this structure here, which we haven't gotten to yet, that allows the saltiness, if you will, to be very general, to pull osmotic force water out of the not the medulla, not the nephron loop, but out of this collecting duct and return it to the body. Now, if we just pulled water out, that's going to mess up our gradient. So the vasa recta help to take that water away. All right, we all obviously want to add it back to the blood anyway. So that's what the vasa recta are doing. They're picking up that water that was pulled out by that osmotic gradient. Again, we'll talk about how that happens, but that's the purpose of having the nephron loop dip down into the medulla, is to create that gradient. Okay? So the distal tubule comes up here between the afferent and efferent arterioles so that the amount of sodium can be sensed, and then it will go into the collecting duct. So this is the end of the nephron. All right? The collecting duct is not part of the nephron. I'm sorry, it's not, yeah, the collecting duct is not part of the nephron structure. So we have an additional tube that starts in the cortex and passes all the way down to the papillary openings at the tip of the pyramid. Multiple nephrons, multiple distal convoluted tubules will dump their filtrate into the collecting duct. So as you look on this diagram, here is the capsule. Attached to the capsule is the proximal convoluted tubule. Dips down as the nephron loop, comes back up as the distal convoluted tubule empties into the collecting duct. Okay? So by the time it empties into the collecting duct, we have removed from it all the sodium, any amino acids or glucose. We've added to it any hydrogen ions or drug byproducts. So we've done everything but determine the concentration. Now, if you drank 2 gallons of water before you came to class today, you obviously don't need to reabsorb all that water. And so when it goes into the collecting duct, it stays in the collecting duct and goes into your bladder as a very dilute urine. If you haven't drunk anything since yesterday morning, your body is dehydrated. So those water channels in the collecting duct will be present, and water will be pulled out of the collecting duct in the medulla and added back via the vasa recta to your body's blood volume. Okay? That is what we're going to talk about on Tuesday and Thursday next week, is how that all occurs. Okay? So, these are the structures, along with the blood vessels, that you need to identify today in lab. And I also, lecture-wise, want you to be aware of what structures and vessels we find only in the cortex, what structures and vessels we find only in the medulla, and what structures we can find in both. All right? So, vessels that we find only in the cortex, of course, the interlobular, the afferent, efferent arterioles, and our glomeruli, and peritubular capillaries, everything that you can see up here. Vasa recta are found only in the medulla. Okay. So here are the vasa recta, found only in the medulla. Peritubular capillaries found only in the cortex. As far as the nephron itself is concerned, the capsule and the proximal convoluted tubule and the distal convoluted tubule are found only in the cortex. The loop can be found in both. If we have a short nephron, the loop will be entirely in the cortex. If we have a nephron near the arcuate artery or close to it, the loop will be found in the medulla. So the nephron loop can be found in either the cortex or the medulla. The collecting duct starts in the cortex, ends in the medulla. All right, so it can be found in both places as well. Yes?
The collecting duct is a vein, right?
No, the collecting duct is a tubular structure formed of simple columnar cells, maybe simple cuboidal here, but mostly simple columnar cells. It receives the filtrate from the distal convoluted tubules, carries it down through the medulla where it can be concentrated or not, depending on the hydration level of the body. So it's not a blood vessel. It's how the filtrate is finally changed to its final composition. And then, this is what is going to, so here's the nephron loop, collecting duct. All the collecting ducts come together to form larger capillary ducts. And this would be the papilla, where it's finally emptying into the minor calyx. And at that point, we call it urine. Do you remember my previous diagram? Okay, so if we take this, so out here, we're going to find the capsule, peritubular capillaries, glomerulus, proximal convoluted tubule, distal convoluted tubule. Nephron loops will dip down in here, vasa recta will be down in here, and the collecting ducts will finally empty right here. These are the openings of the end point of those collecting ducts or capillary ducts finally. And as soon as that filtrate leaves that, it's in the calyx, and we no longer modify it. Okay, we're not going to add water or take out water or add ions or take out ions. Now it's urine. Okay? This is the material that your quiz, lab quiz will be over on Tuesday, kind of like the respiratory system. The next lab quiz will add the histology, what these structures look like in the microscope, because I'm going to talk about the histology on Tuesday, whether it's a simple cuboidal cell or a simple squamous cell, whether it has microvilli or not. And we'll look at that on Tuesday. So your lab quiz on Tuesday is just recognizing these components on the models. All right? So that's the end of lecture. And you take a break. Before you take a break, let me show you the models, and you guys can come and go as you need to. So we have this model showing the muscles, all right? The ureter and bladder we'll talk about histology-wise on Thursday, I believe, of next week. So that part's not going to be on Tuesday's quiz. Tuesday's quiz will stop with these structures here. They're on your list for today, but that won't be on Thursday's quiz. Then we have a couple of models here. So this one, we also have separate kidneys. This one shows the blood supply. So the arcuate, the interlobar arteries, the arcuate arteries, the interlobular arteries. This is looking at one pyramid, so and its associated cortex. So again, this is kind of what I drew on the board. Here's the arcuate artery, interlobular artery. And remember, the nephron is really just a tube. So leaving the capsule is the proximal convoluted tubule. And then we divide the nephron loop into a descending limb and an ascending limb. Okay, so you can use arrows. Descending limb of the nephron loop, ascending limb of the nephron loop, and the distal convoluted tubule. And you can always identify the distal convoluted tubule because it's going to be the one that attaches to the collecting duct. Okay? This structure is what we're going to talk about on a Tuesday in more detail. Here is the afferent arteriole. Here is the efferent arteriole. Okay. Here is the glomerular capillaries. This is the visceral layer of the capsule that's wrapped intimately around the capillaries and the outer layer. And here's the capsular space where the filtrate is first found. And then we have our proximal convoluted tubule beginning. Okay, question?
Yeah, I was wondering, so, actually it's a stupid question. You said we need to know ascending and descending.
Limb.
But vasorectal won't spot.
Well, yeah, the vasorectus is different. Vasorectus is blood vessels.
Oh, yeah, that's right.
Okay? So, this is the same model, it just looks a little bit different. All right? So here would be the vasorecta. Here would be peritubular capillaries. Peritubular capillaries are only in the cortex. Vasorecta are only in the medulla. So this is like one medullary pyramid. Here's the collecting duct forming the larger papillary duct. So this tip right here is the same as this tip right here. So when it drips out of here, it's now urine. Okay? And then the other structure on the model, this is plaster, so it's a little heavy. And it looks scary with all these colors. Okay? But it really is the same. It's just a larger view of the middle portion of that last model. So this is medulla, cortex, arcuate artery, interlobular artery. Coming off the interlobular artery is the afferent arteriole. Coming out of the capsule is the efferent arteriole, which up here is going to form the peritubular capillaries. Down here is going to form the vasorecta. You don't have to worry about these smaller branches, just the vasorecta. Okay? From the capsule, attached to the capsule, you can see the brown structure leaving. So that is brown, yeah, the brown structure. So that's the proximal or tan or biscuit or whatever you want to call it. That's the proximal convoluted tubule. Descending limb. Purple is attached to our yellow collecting duct, so the purple is the ascending limb. And if you're not sure, you can always come up here and look which one's closer or attached to which structure. So purple is distal convoluted tubule, collecting duct, papillary duct. And that's the structure on this model. Okay? So that's the lab activity for today. And what the lab quiz we'll cover on Tuesday. Okay? So if the functions seem all blurry and overlapping, that's what we're covering on Tuesday. We're going to come back to the capsule and start with its structure and what it does. So on those semesters, when I give a separate respiratory exam, I actually overlap those two lectures together. You guys are lucky. We'll go over it twice. Okay? Today, and then we'll go over it again on Tuesday as we add the histology and how it actually does what it does. Okay. How's that like? Doesn't look like she just got her ear. What is that?
I don't know what everyone else was.
1 year of high school. I was certainly starting my freshman year. We had a house, clean the house. All we did was keep our room clean and do dishes. And so we came to the States. My mom started to work. She was an RN. It was nice. So we were informed that I was in charge of cooking. All I'd ever cooked was casserole pudding, and which I loved. And my older sister was in charge of laundry, and my younger sister who was 10 was supposed to help me. So we spent the summer at my grandparents, when my parents were in Maryland finding a house, and I just copied recipes, the entire summer. So I made breakfast, and I made dinner. It was my mother, but she usually would get up from sleeping, eat dinner, and then go back and take another nap. My son did the laundry, by the time, I mean, I just never had any training. I just basically swallowing and, follow the steps. And all of us would, on Friday, we Saturday, on Friday afternoon, Friday afternoon, got to start Wednesday, Friday night. We all had a laundry done and Friday night. And with our kids, we worked in so it was, work after school, come home, do our homework.
I don't know what I'm going to be.
With our kids, it's still regular. There's basketball practice, there's basketball games, volleyball, and so we can always overtime. My husband was 8 o'clock to 10 o'clock, or 5:30 here. So meals are never, on a regular basis, or just extended the calendar. And I try to do assignments, chores for the kids. Half the time we're getting home at 10 o'clock at night, and then it changes. So I'm home last night. 3 nights in a row now. I cooked dinner, actual dinner, a day, quiet dinner on the way home. And I'm doing dishes the day before. I'm cooking dinner. I'm cooking dinner. I said, I'm watching TV. And I'm like this. It's nothing. You should be feeling guilty that I am doing this stuff. And you're just sitting there totally ignoring me. So my youngest son is the best. He will actually on his own when you leave come in and start doing my daughter. My mom does the dishes and I don't have to. That's great. That's like, so we have the entire house being everything. My husband was like a the TGI Fridays. It was just understood. And where did I fail? Training my kids.
He likes my company.
I want to go because, well, the first argument, my husband is great at,
I just finished.
when we were first married.
Really, my sister finished.
He was
Yeah, but I finished it in like 2 days.